Good day, everyone. And today we would like to present to you a presentation about enzymes. So, what are enzymes? Enzymes, or an enzyme, is a compound, usually a protein, that acts as a catalyst for biochemical reactions as defined by Stoker. So, today we have a couple of topics for discussion, and these are enzyme nomenclature, models of enzyme action, enzyme specificity, and regulation of enzymes.
Naming and classifying enzymes. Enzymes are classified and named according to the reaction they catalyze. If the reaction was not specified, such as in lactase, it is highly likely that this is a hydrolysis reaction. Enzymes end with the suffix "ase", and all enzymes end with "in" or "in". These enzymes that ends with "in" are mostly digestive enzymes. So if we saw a name that ends with "asp", it is highly likely that it is an enzyme. So let us be aware of that. And some enzymes, as you can see, they do not have a specific reaction that is attached to them. So we will see these types of reactions and how we will name these and classify these enzymes or identify these enzymes. There are 6 main classes of enzymes, which are oxidoreductase, transferases, hydrolases, diases, isomerases, and ligase. Oxidoreductase. These enzymes requires a coenzyme that is oxidized or reduced as the substrate is reduced or oxidized. So it always comes in pair. There is one that is oxidized and there is one that is reduced. So for example, I have a coenzyme that is reduced and I have a substrate that is oxidized. So if this enzyme is to catalyze that type of reaction, it is called an oxidase, a reductase, or dehydrogenase. So, for example, we have polyphenol oxidase. So, as we can see, we have a substrate that is polyphenol, and we have a coenzyme that oxidates. We have a reduced substrate which is polyphenol, and then oxidases, which is type of reaction that will occur. Or dehydrogenase. We have, for example, we have the lactate dehydrogenase. So we have a reduced substrate, which is lactate, and we have an oxidized coenzyme the enzyme will catalyze into, resulting in a pyruvate, which is oxidized, and then reduced substrate. Transferases. The transfer of functional groups from one molecule to another. I think this one is self-explanatory, and I think all of us are quite familiar with the functional group. So, there are two subclasses, which are transaminase, which is the amino group transfer, and the kinase, transfer of phosphoryl group from ATP to give ATP. So, kinases involves energy. For example, hexokinase. So hexokinase, from the word itself, there is a transfer of phosphoryl group or a use of ATP or energy, and then it transfers hexo molecules. So it did not specify what type of molecule, but it has 6, or it is a pyrran ring. Hydrolases. I think this one should be self-explanatory from the word hydro, which are water. So these are hydrolysis reactions or addition of water to break bonds or linkages. There are 5 main subclasses, which are carbohydrates, which is the breakage of glycosidic linkages that occurs in carbohydrates, proteases, breaking of peptide linkages or amide linkages that occurs in proteins, lipases, breaking of ester linkages in triacyl glycerols, and nucleases, breaking of sugar phosphate ester bonds in nucleic acids, and phosphatases, breaking of phosphate ester bonds. So primarily, all organic compounds has their own enzymes that hydrolyzes them. Isomerase. Isomerization of a substrate in a reaction or rearrangement of atoms in a substrate. First and foremost, there are two main subclasses, racemases and mutases. From the word itself, isomers. So I think this one is of an explanatory. It is a type of enzyme that catalyzes a molecule and thus forming their isomers. So there are two types, which are racemases. Racemases form D-isomers to L-isomers or dextrorotatory isomers to levorotatory isomers. Mutases is a transfer of a functional group from one position to another in the same molecule. So if the transferases transfer another functional group that involves energy, such as kinases, here mutases is only a transfer of functional group so that the molecule will be an isomer of the original molecule. For example, phosphoglyceromutase. So there's a mutase in there, and then the phosphoglycero or the phosphate group and the glycero group or the sugar will be transferred to another position so that it will result into a into an isomer of that molecule. Liase or liase, removing or addition of double bonds. So this might be quite confusing for for other uh for the hydrolysis reactions, but liase does not involve water in it, or uh it does not involve water in it, or it is the addition of a group to a double bond or the removal of a group to form a double bond in a manner that does not involve hydrolysis or oxidation. So we have four main subclasses, which are dehydrates, hydratase, decarboxylases, and deaminases. So dehydrates, it is the removal of water in it. Hydratase, it is the removal of carbon dioxide in it. And the decarboxylases, it is the removal of nitrites in it. And then deaminases, it is the addition of water in it. So, thus, if we remove these molecules or compounds, we will form a double bond, or we will break that double bond. Lastly, we have the ligase. Ligase is also a bonding together of two molecules with the participation of energy or ATP. There are two main subclasses. Synthases forms new bond between two substrates, while carboxylases forms a new bond between a substrate and a carbon dioxide. So there we can identify and class an enzyme.
Model of enzyme reaction. It explains the different enzyme reaction as catalysis in biochemical system. Two concepts to remember: enzyme active site, enzyme substrate complex. Enzyme active site. The active site is a relatively small part of an enzyme structure that is involved in catalysis. So basically, the enzyme active site is a small area part of an enzyme that directly binds to a substrate and carries a reaction. The active site is an enzyme is a three-dimensional structure formed by groups that comes from different part of the protein chain. These groups are brought together by folding and bonding of proteins, basically secondary and tertiary structure. So if you haven't already known, an enzyme active site is actually bonding of many proteins to the point it creates a cluster. The active site usually is a cervic-like location in the enzyme specific substrate complex. And substrate complex is an intermediate reaction species that is formed when a substrate binds to the active site of an enzyme. This is the part when the substrate attached to the enzyme that causes the reaction. Now there are two models that was introduced about the enzyme substrate complex. First one is lock and key model. It's fixed, rigid geometrical conformation, lock concept on certain keys. This model shows one of the enzyme substrate reaction models. The lock and key is a model in which the enzyme is the lock and the substrate is the key. Lock need specific pattern in key to unlock it and fit it order to work. Just like in this model, enzyme is fixed to specific substrate. And another one is induced fitting model. It's flexible, not rigid and static. There is constant change in their shape. Helps to accommodate substrate by a small change in active site. The first model introduced a fixed and specific concept. Induced, fis- induced fit model, on the other hand, compared to lock and key model, is flexible and not rigid. The enzyme is in constant change in their shape, thus are able to accommodate any substrate that enters.
Specificity of enzymes. One of the properties of enzymes that makes them so important as diagnostic and research tools is the specificity they exhibit relative to the reactions they catalyze. A few enzymes exhibit absolute specificity, that is, they will catalyze only one particular reaction. Other enzymes will be specific for a particular type of chemical bond or functional group. In general, there are four distinct types of specificity. Absolute specificity. The enzyme will catalyze only one reaction. For example, urease catalyzes only the hydrolysis of urea. Group specificity. The enzyme will act only on molecules that have specific functional groups such as amino, phosphate, and methyl groups. For example, pepsin hydrolyzes a peptide bond in which amino group is contributed by an aromatic amino acid. Linkage specificity. The enzyme will act on a particular type of chemical bond regardless of the rest of the molecular structure. For example, phosphatases hydrolyze phosphate ester bonds in all types of phosphate esters. Stereochemical specificity. The enzyme will act on a particular steric or optical isomer. For example, L-amino acid can act only as L-amino acid but not D-amino acid. Though enzymes exhibit great degrees of specificity, cofactors may serve many apoenzymes. For example, nicotinamide adenine dinucleotide or NAD is a coenzyme for a great number of dehydrogenase reactions in which it acts as a hydrogenator. Among them are the alcohol dehydrogenase, malate dehydrogenase, and lactate dehydrogenase reactions.
On our last topic, we have the regulation of the enzyme activity. There are two of the following situations which involve the concept of energy conservation. Number 1. It is a waste of energy if a cell that continually produces large amounts of an enzyme for which the substrate is always very low. These situations results as the turning off of the production of the enzymes. Number 2. It is also a waste of energy if the enzyme continues to catalyze the reaction that produces the product. If an enzyme catalyst reaction is present in a plentiful or more than needed amounts in a cell, then the enzymes needs to be turned off too. There are many mechanisms exist by which enzymes within a cell can be turned off and turned on. And within this section, we have three mechanisms that are considered in this enzyme activity. First is the allosteric enzymes. The term allosteric comes from the Greek word allo, it means for other, and for stereos is for site or space. Many, But not all of the enzymes are responsible for this regulating cellular process, as the allosteric enzymes have the following characteristics. Number 1. Because of the composed two or more protein structure, all allosteric enzymes have quaternary structure. Number 2. We have two kinds of binding sites, and that is for the substrate and for the regulators. Number 3. The active and regulatory binding site are distinct from both location and shape. And lastly, we have overall changes in the three-dimensional structure of the enzyme because of the binding of a molecule at the regulatory site. Then we have the regulators. It is substances that bind at the regulatory sites of the allosteric enzymes. As you can see in the picture, the positive regulator inserts to change the shape of the active site for it can be more readily accept the substrate. This increases the enzyme activity. While the negative regulator, after inserting the regulator, it closes the active site and less readily accept the substrate. This decreases the enzyme's activity. On the first mechanism, we have the feedback control. It is a process in which activation or inhibition of the first reaction in a reaction sequence is controlled by the product of the reaction sequence. At the low concentration of the final product, which is the D enzyme, the reaction of the sequence proceeds rapidly, while the higher concentration of the last product or final product, the activity of the enzyme 1 becomes inhibited by the feedback and eventually the activity stops. At the stopping point, there is sufficient D or the final product present in the cell to meet its needs. Later, when the concentration of the final product decreases through use in another cell reaction, the activity of enzyme 1 increases and more final product or the D is produced. Regulators of a particular allosteric enzyme may be products of entirely different pathways of reaction within the cell, or they may even be compounds produced outside the cell or the hormones. Then, we have the second mechanism, the proteolytic enzyme enzymogens. It is an enzyme that catalyzes the breaking of peptide bonds that maintain the primary structure of a protein. It is also based on the production of enzymes in an inactive form that it turns on the appropriate time. Proteolytic enzymes are generated in an inactive form and then later, when they are needed, are converted to their active form. This is called the zymogens. It is also the inactive precursor of a proteolytic enzyme. The zymogen can be also called as a proenzyme. Activation of the zymogen requires an enzyme-controlled reaction that removes some of the part of the zymogen structure. Such modification changes the three-dimensional structure, which is the secondary and tertiary structure of the zymogen. It affects the active site conformation. For example, the zymogen pepsinogen is converted to the active enzyme pepsin in the stomach, where it then functions as a digestive system. Pepsin will digest the tissues of the stomach wall if it were prematurely generated in an inactive form. Pepsinogen activation involves removal of a peptide fragment from its structure, as you can see in the picture. Then, we have the last mechanism, the covalent modification of enzymes. It involves in adding and removing a group of enzymes through the forming of breaking of a covalent bond. It is also a process in which enzyme activity is altered by covalently modifying the structure of an enzyme through attachment of the chemical group to or removal of a chemical group from a particular amino acids within the enzyme structure. The process of adding an ATP molecule in a phosphate group is called phosphorylation, while the removing of ATP molecule in a phosphate group is called dephosphorylation. The protein kinases affect the addition of phosphate groups, and the phosphatases catalyze removal of the phosphate groups. Usually, the phosphate group is added to or removed from the R group of the serine, tyrosine, and threonine amino acid residue present in the protein. The R groups of these three amino acids have a common structural feature, which is the presence of a free hydroxyl group, because the hydroxyl group is the site where the phosphorylation or the dephosphorylation occurs.
So that was everything, and we hope you had a nice time listening to our explanations. And thank you for listening. Have a great day. Bye.